Okay, so this is going to be part 2, and I'll talk about our isolation of a new bacterial species, or we think it's a new bacterial species. So in order to grow the phages, the bacteriophages, you need hosts for them to infect. So the first thing that my students did is we went out to the Penobscot and we took water samples. We brought them back to the lab and we tried to grow up a panel of bacteria. So we could then now have something to try to infect. One of the new bacteria that we found, we characterized more, and this one we named, well, it's temporarily called E2. And this is just because this was the second isolate that we got from Eddington. That's what E2 is. And all of the bacteria that we collected, the students had to isolate it on a plate. And so you grow it on a plate, and this is the bacteria streaked on the plate. You can kind of see what it looks like here, and you get these colonies. And they pick a colony, and then they streak it on another plate, and they pick a colony from that plate and streak it again. And so you streak it multiple times to make sure that it's pure and it's isolated. And from then, you can look at it under a microscope, and that's what we did here. You've got these very small rod-shaped bacteria, and this is a gram stain. It's a test that you can do to kind of generally categorize the bacteria. So some bacteria have a thick cell wall, and other bacteria have a thin cell wall and then an outer membrane surrounding it. And those bacteria stain pink. Other bacteria would stain purple. So these bacteria stained pink. So you see these very small pink little rods. So it's a gram-negative organism, and this means that you've got your plasma membrane, and then you've got a thin cell wall that surrounds it, and then you've got a second lipid bilayer. So you've got an extra layer, and it's a gram-negative cell. And this bacteria, the first couple tests you do a gram stain, and that sort of breaks the microbial kingdom or the bacterial domain into two main groups, your gram-positive and gram-negative. And of course there are exceptions, because in biology there's always exceptions. But that's a good test to start off with, and then you start looking at its oxygen requirements. So we're trying to narrow down what this new organism is. So if you grow it in a test tube without shaking it, you form an oxygen gradient, and at the bottom of the tube you've got less oxygen, and at the top of the tube you've got more oxygen. So you've got more O2 at the top, and you've got less O2 at the bottom. And some bacteria can grow only with lots of oxygen, some bacteria grow only without oxygen, and some bacteria can grow wherever they want, with or without oxygen. And so if the bacteria only grow with oxygen, you would just see cloudiness appear. If they only grew without, you'd only see it down here, but you kind of see cloudiness throughout the whole tube. So the bacteria can grow with or without oxygen. And then a lot of bacteria actually look like this. A lot of bacteria are either going to be circles or rods. And then occasionally you have a spiral thrown in there. But a lot of bacteria really look the same. They either look like little circles or they look like little rods. So how do you tell them apart? Well, we use chemistry. And what we're really looking at is what nutrients go in and what waste products come out. What does it eat? And what does it excrete? And that's how you can actually tell a lot of bacteria apart and identify them from environmental samples or clinical samples. And so we did a lot of different metabolic chemical tests where we see what types of nutrients can it use and what waste products does it produce. And so we've got different plates here with different nutrients in them and different tubes with different nutrients. And then you get different colors based on the chemical activity of the bacteria. So on this plate, it's looking, can the bacteria ferment or break down the sugar lactose? And if they are fecal coliforms like E. coli, on this plate they'd grow metallic green. But we don't have a metallic green there, so it's not a fecal coliform. And on this plate, it's actually looking at a couple things. This plate has sucrose in it, and if the bacteria can break down sucrose, then it produces acid, and that changes the pH, and so you get a yellow color, which ours does. And it also has another test in here, so this is a twofer, where if the bacteria can use sulfate as an alternative electron acceptor, then you get hydrogen sulfide produced, and that forms a black color. So in summary, our bacteria so far, looking at A and B, it's not a fecal coliform because it doesn't produce that green sheen. It's not a vigorous fermenter of lactose. It can ferment sucrose, but it does not use sulfate as an alternative electron acceptor. It does not produce hydrogen sulfide. And then over here, moving on to C and D, so here in C, we're looking at citrate utilization. And again, this is a test based on pH. So you may remember from Gen Bio, citrate is central to the citric acid cycle. And most bacteria really like to use things like glucose to go through glycolysis and then to pyruvate, then to the citric acid cycle. But if you're all out of glucose, what are you going to do? So some bacteria can import citrate and use it as their sole carbon source. They import citrate, or in other words, they bring citrate into the cell and feed it directly into the citric acid cycle. And if they do that, then they change the pH and you get this color here. So our bacteria can use citrate as a sole carbon source. And then D, this is a urease test. So the enzyme urease takes urea and converts it to ammonia. And if it does that, it changes the pH and it turns pink. Well, ours stayed yellow, so it doesn't use the enzyme urease. So we did several other tests, and if you combine all of that data together, you can narrow down the bacteria that you're looking at. And we also looked at the genome. So we isolated the genomic DNA of the bacteria, and then we sent it off for sequencing, and it was sequenced with primers for the 16S subunit of the ribosome. So 16S rRNA, 16S ribosomal RNA gene. And this data, combined with our biochemical data, suggests that we have Enterobacter, a species of Enterobacter. And this was kind of the exciting part, is that it was only 97% similar to the nearest species that were sequenced in the database. And the cutoff for a new species for bacteria is 1.3% divergence. And here we've got a 3% divergence. Now this is preliminary. We haven't sequenced the entire 16S ribosomal RNA gene, just a portion of it. So we still need to get more sequencing data and confirm this, but it's likely we have a brand new species of Enterobacter, which would be pretty cool, and then we could stop calling it E2 and we can give it its own name. Now, like I said, this is still preliminary and we need to back it up, but one of the things that kind of supports we have something new is we did another test where we looked at other sugars that it can ferment, and we grow this in a tube, and in that tube is a liquid with only one sugar. We control what sugar goes in the tube. And then there's also in that media a pH indicator, so if it ferments the sugar, it produces acid and it changes yellow. And there's a smaller tube that fits inside of that that's upside down, and if it produces gas, that little inner tube collects the gas. And gas production, this is something that you guys are familiar with, even if you don't really think about it. Swiss cheese, for example, you get those holes in the cheese because you've got microbes that are producing gas. We also use microbes for fermentation to produce gas for us. Like carbon dioxide gives the fizziness to beer or bread. So the yeast is a eukaryotic organism that is going to produce that gas, that carbon dioxide. So this test right here on this chart, we're looking to see if the bacteria on the first row take glucose and produce acid and gas. And going through all of these sugars, notably, it doesn't ferment lactose very well. And this is exciting because pretty much all other species that are published of Enterobacter, they're all strong fermenters of lactose. So this supports that we might really have a brand new species of Enterobacter. So I'm going to stop here. And then I'm going to move to the new phages that we found. So this was part 2 of 3.